Hello everyone, I'm Jayashree and I welcome you all on behalf of Pulse to the portion of science. This space has been created to keep ourselves updated regarding the various research work that is being carried out by our own colleagues at IGIB. The main objective is to know the various genre of work that is being carried out by our fellow workers and to keep ourselves updated regarding the various scientific advancements in the field of biological research. Another important aspect to this section is to know the process and the efforts that go behind publishing a novel research work. In light of this, for this recent issue, we bring to you the work that is carried out by Partha Chattopadhyay, who is a third year PhD student at Dr. Rajesh Pandey's lab. The title of his work is Long Non-Coding RNAs Harboring Regulatory Motifs Within the Repeat Elements that Modulate Immune Response Toward COVID-19 Disease Severity and Clinical Outcomes. The work has been carried out in hospitalized COVID-19 subjects and it has been published in Clinical and Translational Medicine. Let's hear more about his work from Partha himself. Hello, Parth. I welcome you to the first session of this initiative by Pulse. I would like to congratulate you on behalf of the Pulse team for your recent publication. So we can start this discussion. So can you please give us a brief introduction about the research interests of your lab?
Hello, and thank you for the nice introduction. So the thing is, as a group, what we do in the lab is our lab group works on understanding the integrative genomics of host-pathogen interaction. So in our lab, we understand both the pathogen genome architecture as well as the host genome architecture to elucidate or to understand how during an infectious disease, both the pathogen and host play together to modulate the disease outcome or disease severity. So one of the aspects of our lab research is understanding the non-coding component of the host genome, as we know that is one of the major component of the human transcriptome. So I am working on that particular aspect, understanding the role of non-coding RNAs in infectious disease. And as a part of that work, as part of that particular work, we have we have been working on that COVID-19, and during this time, we tried to understand how the lncRNAs or the long non-coding RNAs are possibly modulating the COVID-19 disease severity. Because the first thing is, as we see that during any particular COVID-19 wave, although the particular pathogen is almost same, there is no change in that pathogen in any given wave, but still we observe a diverse aspect or diverse range of disease severity. So in order to understand what are the factors that are possibly modulating the, so what we observed in this study that there is some prominent role of lncRNAs in which regulates the disease severity. And interesting point or interesting observations from our study was that for example, in COVID, we have multiple grades of severity, like few are mild, few moderate, and few severe, right? So interestingly, in moderate patient, we observed that there was a fine balance between the immune response and inflammatory response, because as we know that immune response helps us to fight the infection, whereas inflammatory response has more somewhat like tissue damage effect, which promotes the disease severity or which accelerates the disease severity. So in moderate group, we observed that particular fine balance between immune and inflammatory response, and a disruption of that balance in that severe group. And at the same time, what we observed that how the lncRNAs are modulating these two response, immune and inflammatory response, across all the severity subphenotypes, as well as in certain unfortunate mortality cases as well. So in this study, we highlighted some lncRNAs like the MALAT1 and some link lncRNAs as well, for which till date we do not have much evidences, but through different analysis, we have highlighted some possible role of those lncRNAs, and we also highlighted that possibly they are modulating the disease severity through multiple mechanisms like the mRNA interaction. And another aspect that I want to mention in this one, that while talking about the lncRNA or the non-coding RNA, we know that they are involved in that gene regulatory function. But
Yes.
at the same time, one of the major feature or genomic feature of non-coding RNAs is means they have higher abundance of repetitive elements in their sequences. And as we know that repetitive elements are one of the major component of the human genome, and they are also involved in that in gene regulatory functions.
Mhm.
So in this study, we identified a new aspect of, or we proposed a new aspect of lncRNA mediated gene regulation through repetitive elements, which at that particular time point was the first of its kind study where we have shown that how repetitive elements present within lncRNA sequences can modulate gene expression and at the same time the disease severity associated with those lncRNAs.
Great. That was a really like that sums up like the work that has been done. So to bring in the next aspect, what was the most like the memorable moment like when you were working on the paper? Like, how did you feel? Like, do you feel like any experiment was very tough, but when you accomplished and you finished that experiment, it was like a big thing done? Was there any such moments while you were working on that paper?
So the thing is, memorable moments, if you ask, not in terms of experiment, because experiment was quite simple, if I can say. But the thing is, analyzing or interpreting the result was the most challenging part. The thing is, see, in the study was focused on the disease severity subphenotypes.
Mhm.
Now, the situation would have been much easier if we were comparing simple healthy versus disease,
Yes.
where we have two very contrasting group and understanding the difference between two contrasting group. But if we take only the disease group and then comparing the subphenotypes, so the contrast between any two subphenotypes are not that much great. So
Yes.
between very two closely related group, understanding the difference between two groups, that was the major challenge in this study, definitely. And hopefully we have I think we have understood through this process that while looking between healthy and disease is important, it is also important to look within that disease to understand the factors that governs the disease severity. So regarding this, we initially at the beginning, very beginning of this study, we used to have a lot of discussion that whether we need healthy individuals or healthy samples in this study or not. And probably that discussion, the hour-long discussion helped us a lot, not only for this study and for other studies as well in the lab. So definitely those discussion points as well as the analyze while analyzing the data. So those points are definitely some of the memorable points from during this particular study.
Nice. And what was the most like that's what was the most scariest moment? Like was it during the working on the paper or like getting the samples? Because clinical study is lot dependent on the sample collection also. So how smooth was it or how was that process of uh, collecting samples and working with the human samples?
So the thing is, fortunately, we do not have that much problem, or we did not have that much problem in collecting the samples because at that time there was a lot of or huge number of COVID-19 cases going on in the city or in the country, if we say. So there, the number of samples or getting the samples was not that much issue, along with definitely the collaborations with good hospitals and all. So sample collection and all that was not a challenge at that particular time. The challenge definitely was that experiment and post experiment, the data analysis and interpretation was the major challenge,
Okay.
definitely.
Okay. So to know something about your working habits, like how much time do you spend in the lab and outside the lab, and how is it in your work timings and everything? Generally in your lab.
So the so the thing is, at that particular time when I was working on this particular topic, so that time COVID-19 was in peak in Delhi, almost in peak in it in Delhi. So apart from working in the topics, we used to have long working hours because of that COVID-19 surveillance work and all. So that time the working hours, there was no limit for that working hours per se.
Mm-hmm.
So sometimes it used to be overnight, working overnight in the lab and the next continue for the next day as well. So we have those phases as well. But at the same time, means while writing or analyzing the data, the situation slightly improved, and then the working or that particular working for that uh, surveillance and those things, that was slightly low, workload was low, so I was able to focus more on this particular work. So the workload was slightly lesser, but definitely not less than somewhat like 14 to 15 hours in some cases.
Yeah, that must have been quite hectic. So
Yes, yes, yes.
yeah. So the next thing is like you must have got like comments from the reviewers after submitting the paper to the
Mhm.
journal. So like just to bring in some like funny element, like was there any experiments that were suggested that you couldn't do and it was unreasonable, or like it helped you a lot in proofreading your paper and making it in a much better version? Like how was that? How was that time of revision?
So during that revision, actually, I must mention that as we know that reviewer two is the most dangerous thing that we always say. So
Mm-hmm.
in my case, reviewer two was in disguise of reviewer one.
Okay.
I received more than 3 pages of comments from reviewer one, while
Okay.
the reviewer two was very nice to the study, and his or her comment was very good. The reviewer two,
Mm-hmm.
reviewer one was extremely critical about the study, and definitely I must mention that the points that the reviewer one asked was definitely valid. So the reviewer one suggested few of certain points that regarding the number of reads and all.
Mm-hmm.
And at that particular time point when I initially I received the comment, so I was surprised, or at the same time I was thinking that how can I or how can we answer all these comments, because some of the comments at that particular time seems like definitely there is no answer, at least at this moment. But
Mm-hmm.
then we worked as a team, and all those comments except for one or two comments we answered. And for those one or two comments that was not answered in that particular sense, not answered in that particular sense, but the thing is we dependent our points there.
Mm-hmm.
Definitely, we tried to accommodate all the reviewer suggestions wherever possible, and of course, especially the reviewer one's comments. So, of course, those comments helped a lot. Definitely, those comments are very insightful, and I was extremely surprised, because what happens ki an experimental person, if the paper goes to an experimental person for review, they will suggest more in terms of the experiments and something. If a
Mm-hmm.
paper goes to some analytical person or computational person, they will suggest more on the analytics part or analysis parts. This time the reviewer one's comment on the experiment and analysis both part was extremely critical, and we all were surprised with the knowledge of that reviewer one that he or she have in that particular domain. But definitely those comments are extremely useful for us to shape that manuscript in the final form.
Yeah. Of course, these constructive criticism from the reviewers is what make a work like much more better, and like it's a better, it
Exactly,
makes it a better
exactly,
version actually. Yeah.
exactly.
So finally, how did you feel when your paper got published? And like, was it like first go it got published in the one journal you targeted and it got done, or was like what was the process behind that final stage?
So definitely it was not a one-shot publication. We
Okay.
initially tried three other journals actually, and those three journals they returned the paper with the comments that the findings are not novel at that particular time. But
Okay.
we had our own uh, defense for that particular comment that the findings are not novel, because we are 100% sure that these particular findings are novel at that time. So though we replied that we defended our point, but still, because this is an editorial process and we don't have that much thing to do if they say that your paper cannot go for review. So in our fourth attempt, we came to that clinical and translational medicine, and the paper got accepted. The point is, if you ask about the feeling when it got accepted, So this is special because of two points. First one is this is my first author research paper, so that is always special.
Yes.
And second thing is the hard work that was gone not by only me but the entire teams, especially during the revision, because during the revision process, another manuscript was in its last phase.
Hmm.
So we have to do two manuscript work simultaneously, almost at the same time, and that was really difficult phase of that work. The more even more difficult phase compared to the analysis and interpretation phase. So completing that particular phase and then getting that success, that is always special. And because of the first reason that I mentioned, that the first author research paper, so it is always special. And I was in between the lab meeting when we received the mail about the acceptance of that paper, and I immediately in between the lab meeting I noticed that, and then I declared, or means I told that the paper is accepted. So yeah, I enjoyed after a lot, and we celebrated the same in our lab as well after that.
Yeah, that must have been really good. Great.
Yes.
So, thank you so much for sparing time, like amid your schedule for this interaction. It was really nice having this, like, it was a very casual, fun interaction. It was very nice to share. Thanks for sharing your experience regarding your first publication and many more to go. Thank you.
Thank you for your last wish, and definitely it is a fun interaction, and I hope that this kind of interaction, not even means beyond this, but platform also goes on between all of our work,
Yes.
all of our fellow members.
Yeah. Thank you so much.
Thank you.